So, if there is any questions or so, I'll be happy to take that.
I'll kick off with a question. I suppose one of the first things, just the way membrane proteins are made, they're quite easy to identify from DNA. So one of the first things from the genome sequencing in the mid-90s was to identify membrane proteins.
Using software that we had developed, actually. So.
Well, there you go. Yeah. And so, and a lot of companies sprung up. I think I raised money for some in the distant past. I apologise for that now. That were targeting particularly things like GPCRs because they're such great druggable targets.
Yeah.
But for some reason, we haven't seen a wave of drugs from that.
Yeah.
Why is that?
I think one, and you can correct me if I'm wrong, but I think one major reason is that they're easy. From the gene sequence, it's easy to tell which genes code for membrane proteins and which don't. So there is very good prediction software that all the drug companies use our software to do these kinds of predictions actually. But that's as far as you get. Then in order to make this a drug target, you have to do the hard work, which is the protein work. It's not enough just to get a prediction. This is a membrane protein. Ideally you need structures of the kinds I showed you if you want to do drug, design-based drug development. And this is hard. This is difficult work, and it's not scalable at this point. So it costs a lot of money. So GPCR, G protein coupled receptors, this big slice of the pie, I don't know how many hundreds of man-years that have been invested in trying to get, or thousands of man-years in trying to get structures for these G protein coupled receptors, and millions of dollars probably, both within private industry and within academia. And it's only in the last 2 years that structures have come out. There have been rumors that commercial companies have been able to do it, but they've never been confirmed. And it was actually done by some academic groups with very large funding.
Which protein was that?
Sorry?
Which protein was that in specific?
So it's the first one, I think, was the beta-adrenergic receptor. Beta-adrenergic receptor. Adrenergic, yeah, it's from heart muscle, I think. So, yeah. I think that was the first one, and then there's one or two more. There is also one related protein from our eyes. So there is a G protein coupled receptor, or the light-absorbing protein in our eyes are G protein coupled receptors called opsins, and they were crystallized a little bit ahead of the other ones, and structure solved. But they're totally uninteresting for drug development because they don't have obvious diseases associated. So I think there are maybe 2 or 3 types of G protein coupled receptors that have been solved where structures are known now. But this has been a massive effort, from academia and industry over a very long period, probably 15 years or more, to get these structures. So they have been extremely difficult to get.
Just in that respect, do you think that's now been cracked and you can now just turn the handle and standardize it?
You cannot just turn the handle, but people are getting better at it. And they're getting it in active and inactive forms and stuff like that. So there will be I don't know if you can say a steady trickle, but there will be a steady trickle of new structures coming over the coming years, I think, because they have found some kind of practical way that pays off in terms of attacking these structures.
What about sort of therapeutic areas that you're thinking of then?
So there's probably other people that know better what the therapeutic. I mean, I think a lot of drugs that you use to treat psychiatric conditions actually have G protein coupled receptors as targets. So, I mean, the drugs that are currently sold, of course, were done by classical, drug development techniques, screening, without having structural information. But they only touch a small number of GPCRs. There's like 700 or 800 of them, different ones in us, and the known drug targets are maybe 3 or 4. I don't know, maybe a handful. So.
Pfizer built this big facility in Kent that was basically just screening for GPCR proteins and trying to find drugs. Now it's gone, but that was basically only for GPCR proteins.
But of course, like I said, there are many other membrane proteins like ion channels and various other transporters and things which are good candidate drug targets.
What were you saying? Sorry, GPCR, you're talking about a specific kind of membrane protein.
It's a group, GPCR is a group of proteins that, I don't know, you had the percentage, how many of the membrane proteins?
I think total of all proteins in humans, in the human body, 5% are G protein coupled receptors.
Those were considered as the hottest kind of targets for drugs because because
they're involved in so many different signaling events. You know, they sit on all kinds of cells and they receive signals from the outside in various kinds of signals. So they're a very easy entryway into cells if you want to to affect what's going on inside the cell. If you can block or activate one of these receptors, it's a very easy way to get into the cell or to manipulate what's going on inside the cell. And they're involved in all kinds of biological functions in our body. So it's